Right. Okay. Well, so here is a zebrafish. I'm going to talk about it in more detail later on. Um, so I switched the screen. Can you see the second slide?
Yes.
All right, good.
We see it.
Yes, thank you. So zebrafish is currently second after mice most used in biomedicine based on the number of animals used in research. As a model species, it has multiple utilities, multiple um, aspects that allow us to use it as a model species. It's relatively inexpensive. It allows the whole animal assays, the cellular assays, the availability of whole body, whole organism imaging, the homology of genome and physiology, the conservation of molecular targets. All that is really making zebrafish a useful model in biomedicine. Now, compared to mice, Drosophila, zebrafish, in fact, becomes the most useful model organism in biomedicine. So today I'm going to talk about the most useful model organism in biomedicine, which is currently number 2 most used animal species in biomedicine. And time will pass, and we will see that zebrafish becomes probably number 1 rather soon. So it's a very important model organism, and the question is, how can we use it to solve complex biological problems? Right. Okay. Just to show you the number of publications using zebrafish medicine is really skyrocketing, and different countries are major players in the field. And for example, Russia has some input, which in my view needs a lot of work so that we are on par with other countries in this race. Zebrafish is a useful genetic machine because it has 70% homology with human genes. And interestingly, about 85% of human disorder-related genes have their orthologs in zebrafish. So when it comes to diseases, the homology of humans and zebrafish is even higher, 85%, which is quite impressive per se. The question is why we use zebrafish, and the simple answer that I want to give is that. Now, zebrafish allows us to move the time frame of the natural evolution from 70 million years ago, when the first primates emerged, to 300 million years ago, when the first mammals emerged. And again, zebrafish is much older than mammals. So we're moving the time frame of the natural evolution to 400 million years ago. If in the process of our research, we are lucky to establish an important pathway that has conservation between the 3 species, between the humans, the rodents, and zebrafish, as shown on this slide, chances are that we are really targeting a very important pathway that is in the center of the pathogenesis, in the center of the pathology. That way, we can target that pathway, its targets, and eventually treat the disorder. Just a few examples from research from my labs. showing the utility of zebrafish in neuroscience. Here is an example of applying a chronic stress protocol to probe the brain genomics, identifying several important clusters of telencephalic genes that are differentially expressed in zebrafish, including arrestins and GPCRs, that is cluster A, the genes related to ubiquitination, cluster B, the inflammation-related genes, that is C cluster, and the cytoskeletal genes. So zebrafish data allow us to understand and appreciate the importance of several clusters of cellular genes, such as cytoskeletal and ubiquitination-related genes. So that gives a new appreciation to several potential important biological processes that occur as we apply chronic stress to a complex organism, such as zebrafish. Another example is the use of laser that we developed in my lab to induce the traumatic brain injury. And the advantage and the beauty of zebrafish model is in the transparency of the skull and the skin. So the laser can penetrate inside the brain and through the skull without any damage, no infection, nothing. So you can really very targetedly induce the brain trauma. And then, of course, we got to use different types of tracers to see the expression of certain important genes and proteins, as is shown on this slide in case of BDNF, which is an important brain neurotrophin, and we can trace its expression in telencephalon across the days after the trauma in order to understand the recovery and the rescue. Now, I'm going to the field of drug discovery and drug screening. And importantly, zebrafish is an excellent model for drug screening. There are papers showing that you can screen up to 10,000 drugs a day using zebrafish. Well, that was done in larval fish, as shown on the left. Under the microscope, you can put a small larva into each of the 96 well plates, and you will record 96 zebrafish, each given a specific drug. So 10,000 drugs a day sounds like a feasible undertaking. For adult zebrafish, it's much slower, of course, but you can still test hundreds of drugs a day. In my lab, we have developed a new model of a new method of three-dimensional phenotyping of adult zebrafish when traces of zebrafish influenced by different drugs and experimental manipulations are recorded using a 3D approach, so that we can recreate the 3D traces of zebrafish and then decode those traces using computers to identify specific phenotypes that we are interested in. And as shown here in the middle, a gallery of different traces induced by various drugs. So, for example, caffeine, chronic morphine, nicotine acutely, but also withdrawal from drugs that cause addiction. Some drugs that are important medicines, such as chronic fluoxetine, and some genetic manipulations as well. So three-dimensional structures of drug responses are really important and interesting. Here is an example of what nicotine does to zebrafish. On the upper right corner, you see a real three-dimensional response of nicotinized zebrafish. So as you could see, zebrafish is swimming in stereotypic patterns at the top. That means it has no anxiety. It's an anxiolytic drug that has psychostimulant property. Now, in the middle, on the upper half, you can see the control zebrafish that shows nothing similar to nicotine at all. Here are a few examples of drugs for which we created the libraries of three-dimensional reconstructions. The controls is on the left and caffeine is on the right. You see, an anxiogenic um, drug in zebrafish, and so is in rodents, by the way, and in humans. If you drink 9 cups of coffee, you will have panic attacks. So zebrafish are highly sensitive to such drugs. Now, on contrary, chronic morphine and chronic ethanol produce a totally different picture. Zebrafish are very active. They swim slowly but surely. And this is obviously very different from both caffeine and control groups. Now, all that has been widely published and widely cited. And quite recently, we made another effort. We created for the first time the artificial intelligence-based platform to use the library of three-dimensional phenotypes that we have accumulated to train the neural network to recognize the patterns based on the known pharmacology of those drugs. So, for example, the morphine and all the opioid ligands will produce similar phenotypes, and the system will learn from these phenotypes, but it will also learn from nicotine and nicotine-like drugs that cause a different three-dimensional response in live zebrafish, and it will learn from this class of drugs, and then it will move to ethanol, and it will learn how ethanol does things to zebrafish, and each class of drugs is somewhat similar but somewhat different, and the artificial intelligence is intelligent enough to pick up this subtle differences. Now, here's an example of how that works. So after training of our network using a library of nearly 30 major neuroactive drugs, we found that the artificial network can distinguish, for example, different and can cluster different drugs based on their chemistry. So, for example, it would place ketamine, which is a glutamatergic, anti-glutamatergic drug, separately from, for example, monoaminergic drugs like mescaline and psilocybin and cocaine. It will cluster psychedelic drugs such as LSD and MDMA separately. And then, sure enough, these are similarly acting drugs. And in fact, it will predict MDMA as MDMA with 71% accuracy. And it will also predict highly significantly that LSD and MDMA are very similar. When we developed this platform, we wanted to verify using a new drug that this platform didn't know about. So we used aricolin, which is a cholinergic drug. It's similar to nicotine by chemical formula, but also in physiological properties. Now, the system predicted aricolin and nicotine as similar with the 80% probability. So because it has never seen aricolin before, it thought it's nicotine. But in fact, it's a very similarly structured drug that is similar to nicotine, but it's a different drug. But 80% accuracy of prediction is high and promising enough for us. The next level, the next step that we made was to apply the system, the artificial intelligence powered platform, to test a battery of 10 novel N-bones, the derivatives of benzyl phenylethylamine that have multiple substitutions in two moieties, the phenylethylamine moiety and the phenyl ring of the benzyl moiety. The structures are shown on the slide. There are different drugs, as you could see, um, um, produced in collaboration with chemists from Kazan, totally new. These drugs have never been tested in any living system before, and we were lucky to try and see what these drugs do to zebrafish. On the right half of the slide, you can see behavioral effects of some of these drugs. Some of them cause increased anxiety and hallucinogenic effects. Some of them are behaviorally inert. Some of them cause anxiolytic properties, and some of them are very potent hallucinogenics. So all this was quite predictable. We also studied neurochemistry of these drugs, looking at the expression at the levels of serotonin, dopamine, and norepinephrine and their metabolites, identifying, as I mentioned, four clusters: anxiogenic, behavioral inert, anxiolytic, and anxiolytic and hallucinogenic compounds. Now, based on the artificial network, intelligence network that we created, these drugs have been predicted as having serotonergic and antiglutamatergic properties. We then applied a chemical modeling approach using the PASS online software that Professor Perret and his colleagues have created to independently run these drugs and their formulas against the known biological mechanisms and cascades that might be related and activities, identifying again serotonergic and antiglutamatergic properties of these drugs. In other words, we started with testing real drugs in a complex in vivo system. We analyzed and created libraries of these responses, used artificial intelligence to predict the pharmacological properties. We then used chemical modeling, such as PASS online, to predict their pharmacological properties based on chemical formula. We got a perfect match. So our behavioral, pharmacological, neurophenotypic data confirmed the prediction made using the chemical formula. And then we made our conclusion that these drugs, new drugs, may have two important functions in biological system. So I will conclude by saying that zebrafish provide an excellent platform for in vivo drug screening. They produce high throughput, automated, unbiased analysis for drug-induced responses in vivo, and they allow us to predict the drug pharmacological activity with a relatively high accuracy. At this, I will stop. I thank the organizers and the audience for having me here, and I put my email address in case if anyone has questions, because I knew I will consume all my time and I will leave no time for questions. So here is my email. I will be happy to answer questions.
Okay, thanks a lot, Allan. And you really used all time that was allowed. Yeah, but we have the email from the next speaker that due to some unexpected situation, he could not present his talk in time. So we have 20 minutes for discussion. Yeah. Maybe there will be some questions in the chat, but maybe I start just from myself. And first question is that you mentioned that evolutionary zebrafish is older than mice and than man. And you mentioned that it may be some diseases which are the same, despite the many years and many thousand years of evolution. So we know the human disease ontology. There is also some ontology on mouse disease. What about the ontology of zebrafish disease?
Well, this is a good question. I would say, as a physiologist, that a human disease may not be equal to a zebrafish disease. What we're talking about is the fact that diseases evolve. So a disease that exists in zebrafish may not be the same as we see it in mice. Here's a very simple example, which might be interesting to mention. So, for example, humans and mice have very clear-cut anxiety and depression-like conditions. So we can tell when mice is anxious or when the mouse is sad, and in humans as well. Well, in zebrafish, based on drug responses, we cannot tell that difference so well. So anxiolytics actually work as antidepressants in zebrafish, and antidepressants actually work as anxiolytics in zebrafish. This raises a question of whether in zebrafish there is a simpler syndrome, let's call it affective syndrome or anxiety depressive syndrome, that is not yet dichotomized into anxiety and depression that we see in humans. So I want to say that, again, human disease may not be the same as a zebrafish disease, but the common mechanisms and the shared features may be present. So, and of course, that complicates a lot the composition of the ontology of zebrafish disorders, because we need to study zebrafish more and more to be able to tell what is the disorder in zebrafish. On top of that, as you, of course, know very well, zebrafish have genome duplication, which means that there are certain genes that exist in zebrafish in two copies, about 25% of those genes. So for some important proteins, humans have only one copy and zebrafish have two. So that, of course, complicates the cross-taxon comparisons enormously. But that is to say, we need to study zebrafish more.
Okay. Another question is the dose. Because you told that one could study 10,000 drugs per day in zebrafish. But what is the criteria to select the appropriate dosage or concentration? How do you guess which amount of drug or pharmaceutical substance you should add to get the results? If some dose response curves sometimes were created for zebrafish, at least for some drugs.
Right. Well, I mean, if you can test 10,000 drugs a day, that also means you can test 10,000 doses a day of one drug. So you keep your lab busy, you give tasks to your PhD students, and everyone is happy. Now, of course, we can create the dose dependence curves, and they're pretty similar in many ways to what we see in mice and in humans. And this is very important. But you asked me about the first guess. And here is my answer after studying many drugs in zebrafish. We, of course, give drugs via water immersion. That's the most commonly used method of drug delivery in zebrafish. So you don't have to inject the fish, you don't have to do anything. You just simply put zebrafish into water with the drug. And if drug is not water soluble, you can use some detergents in small doses like DMSO to dilute the drug. And at these concentrations, the DMSO and other um, chemicals will not affect behavior or physiology of zebrafish uh, big time. Yes, and through the gills, which are full of blood vessels, the drug easily penetrates into the system circulation, and it's in a way similar to intravenous drug administration to humans or mice. So you have a lot of drug quickly penetrating into the systemic circulation. Not to mention, of course, some percent of drug absorbance through the skin, which also occurs in zebrafish. So we quickly deliver the drug into the circulation in zebrafish. And if you ask me if I study a drug that I don't know, what I will do is I will look at clinical data, I will look at mouse data, I will get the doses used in mice and humans expressed as, let's say, milligram per kilogram of body weight. I will then assume that 1 kilogram of a body weight equals to 1 liter of a water that we place the zebrafish in. So my first guess will always be I will use the human dose or a mouse dose that works, convert it into milligram per liter, and give it to fish. I will then see what happens. And if that drug kills the fish, I will take 10 times less, a lesser dose. And if it doesn't do anything, I will increase the dose 10 times. And then I will use the increments and I will move left and right along the doses axis to see whether I'm lucky to find the correct dosage.